Assalamualaikum warahmatullahi wabarakatuh. This is new chapter, plants reproduction and growth. So these are the syllables. So we will cover on asexual reproduction, the sexual reproduction, which include the structure of flower, pollination and double fertilization, and primary and secondary growth. Okay, for the learning outcomes, at the end of the topic, you should be able to define asexual reproduction in plants, describe the type of asexual reproduction, and state the type of vegetative structure. So these are the learning outcomes that we will cover on this video. Okay, so for asexual reproduction in plants, many plants are able to propagate themselves using asexual reproduction, meaning this plant doesn't need sexual reproduction in order to live or to reproduce. Okay, so the definition for asexual reproduction is the production of new individuals or offspring whose genes all come from one parent, meaning if the plant is undergo asexual reproduction, there is no difference in their genetic material between the offspring and the parent. So this process involves no fusion of eggs and sperm, so does not involve any fertilization between the male and female gametes, and it relies entirely on mitotic cell division. All the offspring or progeny are genetically identical to the parent and to each other. So it's like clone. Okay, so the production of offspring from single parent and occurs without genetic recombination. Recombination means the interference or the combination of the male and female parent, okay, to produce the offspring. So there is no genetic recombination and they result in a clone. The offspring and the parent are similar or identical. So there are several types of asexual reproduction. Apoxymis, which includes some citrus plant like oranges, okay? Certain grasses such as Kentucky bluegrass and dandelions. The other is natural vegetative propagation, artificial vegetative propagation. From the word artificial, it means it involved with the human activities. Sporulation, the dispersal of spores, normally happen among mosses and ferns, and the binary fission among the algae. So let's see on apomixis. Apomixis is a form of asexual reproduction in plant which seeds are produced without pollination or fertilization. So a diploid cell in the ovule, okay, diploid cell in the ovule in the female, okay, give rise to an embryo. So the ovules mature into seed which then disperse. The advantage of apomixis over other methods of asexual reproduction is that the seed and fruit produced by apomixis can be dispersed by methods associated with sexual reproduction. For example, some citrus or certain grasses and dandelions. So these are the apomixis, where the seeds produced without the pollination or fertilization. Okay, for vegetative propagation, is a form of asexual reproduction in plant where the new plants are produced from vegetative part. So you must know what are the vegetative parts. Okay, in vegetative production, there is no embryo produced. Okay, it can occur through fragmentation or regeneration of a plant part. For example, the stem, the leaf. Okay, so stem and leaf are a part of the vegetative parts. And the growth of specialized vegetative, which is non-sexual plant part, for example, the bulbs, the runners, and the tubes. We will see the image of this part later. Okay, so these are the vegetative propagation through the fragmentation. Okay, fragmentation is the separation of a parent into parts. Okay, from one parent, they separate into several parts. So each of the part will develop into new organism. So that is the fragmentation. So it is common form of vegetative production. The root system of a single parent gives rise to many adventitious shoots that become separate shoot system. So this is the process of how fragmentation happen. Okay. Vegetative propagation can occur naturally or artificially. Fragmentation happens naturally when small part of plant fall off onto the soil and begin to grow into a new plant. Okay, for example, fragmentation occurs in liverwood plants whose small stem or leaf. Okay, the stem or leaf are often broken off by animals or wind and then grow into clones, okay, of the parent plant when they land in the soil. So this is one example how the vegetative propagation happen in natural. Okay, fragmentation happen artificial. Okay, when the human involved with this process. Okay, when we cut some parts of the plant, for example, the stem of the plant, and then we plant it into another place. So that is another process of asexual reproduction by interference of human activities. So we call that as fragmentation. For example, gardener taking a cutting, okay, cutting stem or leaf from a plant. Okay, so these are the vegetative structures, okay, that involve in asexual reproduction. We have bulbs. Okay, we have rhizome. Rhizome is the stem of the plant, but the stem is underground. So when the stem is found underground, the name is no longer stem, but we call it as rhizome. Corm. Okay, the runner or stolon, the taproot, the suckers, the stem tubers, and the root tubers, the leaf, and the stem. Okay, so what is bulb? Okay, bulb is thick. So this is the character of the bulb. Thick and short, which found underground. Okay, so bulb is the underground stem for food storage. So this is bulb. The example of bulb we can see in tulips and onions. Okay, the rhizome, horizontal underground stem for food storage. Example, the ginger. Okay, so this is the stem segment, but this stem is found underground. Okay, so we call it as rhizome. Okay, the runner or stolon. So this is the runner or stolon. Okay, the horizontal stem that grow along the surface of the ground. So normally this stolon found on the surface of the ground, not underground, on the surface. Example is strawberry. So the taproot. Okay, so we have learned previously that taproot, we have two types of root, taproots and the fibrous root. But the vegetative structures for taproots is referred to the swollen food storing. Okay, example, the carrot. So the carrot, the structure of the carrot is like a taproot structure. Okay, alright. Then tubers. Okay, tubers is the enlarged or swollen ends of horizontal underground. It can be swollen ends of stem or the adventitious roots. If it is the swollen ends of stem, we call them as stem tubers. If it is the swollen ends of adventitious root, we call them as root tubers. Example are potato, yam, sweet potato, or the cassava. Right, so this is the root tubers. Okay, and this is the stem. Okay, some of them form stem tubers, some of them form the root tubers. Okay, so this is corm, another vertical underground stem. Okay, vertical means the position of the corm is upright. Okay, not horizontal. Horizontal is like this. Okay, it lies. Okay, so the example is cocoyam. Another vegetative structure is leaf. Leaf, they form buds at the edge of leaf. Okay, so this is the leaf bud. So later on, this leaf will fall on the soil. Okay, and then they form another new organism. Okay, example of the organism is bryophyllum. Okay, so this is the genus of bryophyllum. Okay, next is sucker, young shoots that arise from underground stem. So this is the ground. Okay, so underground there is stem underground. Okay, so this sucker develop from the underground stem. So we call them as sucker. Example of organism: banana, sugarcane, and pineapple. Okay, so the mechanism of asexual reproduction for artificial vegetation. So this artificial vegetative is involved with the human activities, and the common methods usually used is by cuttings, by layering, and by grafting, and also by micropropagation. So micropropagation will involve the tissue culture method. Okay, so the methods of artificial vegetative propagation, the first one is by cutting, where the plant part is cut and planted in other area in the soil. Okay, this cut part will develop into a new plant. Okay, so example, we do stem cutting for sugarcane, Duranta, Coleus, rose, China rose, and we do root cuttings for tamarind, for lemon plant, and leaf cutting for peperomia, begonia, and African violet. So these are the example of stem cutting. We cut from the stem, okay, and the root cutting and the leaf cutting. Okay, sometimes we do leaf cutting for aloe vera, right? So from the leaf cutting, we plant them into a new place. Okay, another is micropropagation or test tube cloning. We do test tissue culture. So this is genetically engineered process, okay, where we use the tissue and cell culture technique. So how we do this micropropagation? Of course, we must conduct this experiment in the lab in a sterile condition, okay, because these tissue are very fragile, okay, and they need a very sterile condition in order to prevent from any bacterial infection. Okay, a small piece of tissue is excised. It is taken from plant and grown in a nutrient medium. We call media. So that media is the nutrition for the growing plant. Okay, under aseptic condition, meaning very sterile condition, the tissue will proliferate. Okay, proliferate means start to grow into an undifferentiated mass called callus. Okay, the first tissue that develop, okay, from the tissue that we cut, okay, that we excise from plant, we call the callus. Okay, so callus is undifferentiated mass, like stem cell. Okay, so this callus can be kept and multiplied for an unlimited period. So we can, once we get the callus, we can also grow another plant just using this callus. No need to excise from the plant. Okay, so when we get the callus, we just take the callus and then plant to another petri dishes. So callus can be a stem cell to grow the plant, beside taking another tissue from the plant. Okay, so when small portion of the callus tissue are transferred to another medium with specific hormones, the differentiation of plantlets is induced. Okay, the plantlets are transplanted in pots or soil and raised to maturity. Okay, so this is the example. Normally we do tissue culture for plant that is exotic plant, or plant that is having species or going to be extinct, or very valuable plant. Okay, normally orchids. We do tissue culture on orchids because some of the orchids are rarely to find in the woods. Okay, and they are very precious. Okay, by this method an indefinite number of plants can be obtained, so we can get this plant in numerous number by doing tissue culture. But tissue culture is a very expensive procedure, and to maintain it we need very high cost. Okay, so to use tissue culture technique, we must make sure that the plant that we culture is very valuable. Okay, very valuable in terms of money, and also in terms of the environment sake. Okay, so these are the environment in tissue culture room. Okay, so we need to switch on the air condition 24 hours. Okay, so this is the media, the agar media. Okay, in this agar media, we put all the nutrition and the hormones, the vitamins that the plant needs. Okay. Okay, so these are the advantage and disadvantage of the vegetative propagation. Okay, so we know that the advantage is we can produce little or no viable seed. Okay, plant that for propagation of plant that produce little or no viable seeds. Okay, so for the plant that is have low production, or we want to maintain the seeds variation. For example, we have one very good seed. Okay, so we want to maintain the genetic inside the seed, so we can carry the vegetative propagation, because the progeny and the parent are genetically identical. And the second is to provide consistent quality without variation. So this is very important if the person is doing agriculture. Okay, so we need a consistent quality of production. This can help the plant to spread and enable survival following damage to parent tree. For example, the parents are damaged, will still have the genetic of the parent. Okay, so that are the advantages of the vegetative propagation. The disadvantage, this plant, okay, which undergo vegetative propagation, more susceptible to disease, because there is no genetic variation. That's why genetic variation is sometimes quite important in order to counter the disease. Okay, and then the second disadvantage, no new varieties can be produced from the process.